Welcome back to Lecture 5, everyone. We're going to continue talking now specifically about chemical synapses in terms of how they release their neurotransmitters and then how the neurotransmitters then give a signal to the postsynaptic cell. So if we kind of look at how this fits into the overall outline, we just did a quick review of action potentials and types of synapses. Now we're focusing on those chemical synapses, and we're doing 2 of the 3 final sections. Neurotransmitter release. Here we go. So neurotransmitters, those chemicals, those molecules that are going to send our signal to the postsynaptic cell, to the postsynaptic neuron, are in vesicles, which are little membrane-bound packets, right? So they're in vesicles. You can see this synaptic vesicle here, right? So synaptic vesicles, and these are located in different places within the neuron, and the book talks a lot about this. I'm not as interested. So they talk about the readily releasable pool, the recycling pool, and the reserve pool. I just want you to know that some are really close to the membrane, they're ready to go. Some are relatively close to the membrane, but a little bit farther away, and then some are pretty far away. So that's going to become important later on when we want to understand neurotransmitter release for different types of pathways and how sometimes it's really easy and quick to get those neurotransmitters out there, and other times it takes a bit more work. So if they're farther away, it's going to take a bit more work for us to get the neurotransmitters out. So just keep that general concept in mind. So when the action potential arrives, so we have those opening of sodium gates, sodium rushes in, changes the membrane potential. It's a big electrical signal that travels down the axon. When it arrives at the axon terminal, calcium channels are going to open. So they're voltage gated, so the change in voltage of the action potential causes these ion channels to open. So then calcium is going to move according to its constant electrochemical gradient, which in this case, calcium is going to move inside the cell. So let me change here. So calcium, can I make a Ca? Kind of, sort of, right? So calcium is going to move inside the cell. I meant to use purple so that it matched, but I didn't. Now, that calcium present inside of the cell is then going to trigger the next series of actions. Calcium is going to enable something called the snare complex of proteins to interact and result in exocytosis. So what we have, let me change to purple. Over here is a collection of proteins. One type is attached to the vesicle itself. That's often called a V snare. And then we have these other ones down here that are attached to the plasma membrane, which is often called the target membrane. And so those are referred to as T snares. I am not interested in you memorizing the names of these proteins, nor which ones are V snares or T snares. What I want you to know is that there are these proteins that when calcium comes into the axon terminal, these proteins interact with each other in a way that pulls that vesicle, that synaptic vesicle full of neurotransmitter down so that it becomes in contact with the plasma membrane. Now the vesicle had a membrane of a phospholipid bilayer, just like the cell membrane does. So when they come in contact with each other, they fuse and then they can open. And what that allows to happen is the neurotransmitter, in this case it's glutamate, that's why that you see that glue. The neurotransmitter then is free to diffuse out into the synaptic cleft via the process of exocytosis. So you might remember exocytosis literally means exit from the cell, exocyte, leave the cell. So the neurotransmitter is going to just, now it's like the door's open and it's going to drift out into the synaptic cleft. So again, calcium is what triggers that snare complex to pull the vesicle down to the membrane where it fuses and releases the neurotransmitter into the synapse. So action potential arrives, voltage gated, I should go this way, voltage gated calcium channels open, calcium moves in and activates the snare complex of proteins. The snare complex pulls the vesicle down to the membrane and the neurotransmitter exits the cell via exocytosis and into the synaptic cleft. All right, now that the neurotransmitter is in the synapse, what happens? So it's going to bind to receptors. So it's just diffusing out into the synapse, passive process, right? And oh my goodness, somebody's such a pest today. And it's going to bind to receptors on that postsynaptic cell, on the cell that we're trying to signal. Now, all right, the screen might jiggle around a little bit because he's rubbing against it. So what I want you to know about receptors is that receptors are specific to the neurotransmitter. In that last example, they had glutamate as a neurotransmitter. Glutamate will only bind to glutamate receptors. Acetylcholine is a neurotransmitter. It will only bind to acetylcholine receptors. Dopamine has dopamine receptors. Serotonin has serotonin receptors. And not only that, but sometimes you can have different types of serotonin receptors that are specific only to serotonin. So just remember that the receptors are specific to the specific neurotransmitters. You can't bind to somebody else's receptor. When we're talking about types of receptors, then, we care about what type of neurotransmitter they bind to. Is it dopamine? Is it serotonin? Is it GABA? Is it glutamate? Whatever. And then they can be of two different classes. They can be ionotropic, and the key there is that the word ion, isn't it? My cat, stop. Sorry, he's making things pop up on the screen. Go away. Or are they metabotropic, which is a really weird word. So let's take a look at those. Ionotropic receptors are literally ligand-gated ion channels. So way back we learned about the 4 different types of ion channels, and one is ligand-gated, which means that when a molecule binds to them, they open. So in this case, ionotropic receptors are ligand-gated ion channels where the ligand happens to be the neurotransmitter. So in the Limb book, they have a diagram here, which I think is a little confusing because they're showing you 2 of them, and it's not a before and after. It's just a like happening all at the same time. But so let me point out what's going on. So the little red molecules here are neurotransmitter that has been released into the synapse. And then you can see, I think I'm going to switch to the highlighter. You can see right here that the neurotransmitter has bound to both of these ionotropic receptors, which have then opened, and the purple is supposed to be ions, and so now the ions in this case are moving into the postsynaptic cell, right? This is all in the postsynaptic cell. So depending on what type of ion channel it is, what type of ion is moving, and in what direction, it's either going to move the postsynaptic cell either closer to threshold or farther away from threshold. So it's either going to be an EPSP or an IPSP, right? So then all those things, the summation and all those things then will affect whether that neuron then sends a signal by firing its own action potential. So ionotropic receptors are really cool. They're really fast. They're really short acting. We're just going to cause little changes in membrane potential, EPSPs and IPSPs, as a result of those ion channels opening. Metabotropic receptors are different. So they're also called G protein coupled receptors, which is, it's all a mouthful, I know. So because, oh, I thought I turned my highlighter on. Yep. Because there are these little G proteins, you can see that here, that are attached to them. That's kind of annoying. I'm going to erase it. Okay, so when a neurotransmitter binds, again, the neurotransmitter is being shown here in red. So this is the neurotransmitter. When it binds to the G protein coupled receptor or the metabotropic receptor, this G protein then causes a bunch of things to happen, which result in another molecule being formed, which is called a second messenger. There are different types of second messenger molecules that can be formed, and they're called second messenger because the neurotransmitter was the first one that came in, bound to our metabotropic receptor, and then as a result of that, the metabotropic receptor is going to make another messenger, which is going to go out into the cell. So that's our second messenger. Now, what this diagram from the Limb textbook is showing us is that that second messenger then is causing a separate transmembrane protein, an ion channel to open, and then we've got some ions moving into the cell. that might be an action that happens as a result of a metabotropic receptor, but they have lots of other possibilities. So these G protein coupled or metabotropic receptors can also, that second messenger might go open an ion channel, but it also might go into the cell and change how proteins are being made and structured and behaving. It can also go and tell the nucleus to transcribe different genes or to not transcribe them, which then results in changes in proteins in the cell, which changes the behavior of the cell. So these guys are really diverse in terms of their potential effects. They are slower onset, right? Because it's like, we got to make this second molecule, then we release it, and then it goes to this other part of the cell, and it tells it to do something. So this is slower onset, but can't be very long lasting. So this is a very different type of way of having a signal come into a cell to change its behavior. So in summary, neurotransmitters then diffuse into the synapse and bind to their specific receptors that are specific for that neurotransmitter only. If it's an ionotropic receptor, that is a ligand-gated ion channel, it's going to open the ion channel. Whatever ion it's specific for, then is going to move according to its electrochemical gradient, causing an EPSP or an IPSP, stuff you've heard about before. If it's a metabotropic or G protein coupled receptor, then it might also, through that second messenger, open ion channels, but it might change some proteins inside the cell, or it might actually change gene expression. So it has a diverse array of options depending on the specific receptor that it will do. So a lot of the book talks about some different pathways and different things that happen. We're not going to focus on that now. We're going to come back to that later. But just keep in mind that overall, if you end up opening ion channels, then you're getting movements of ions, and so you're getting changes in membrane potential, which are going to be either EPSPs or IPSPs. If you are affecting a metabotropic receptor, however, you also have the other options of changing proteins and gene expression, which basically is changing the cell behavior in a way that's different from whether or not it's going to send an action potential or not. So this opens up a whole potential of lots of things that we can tell other cells to do, which is pretty cool. So in the last part of lecture 5, we'll talk about what happens to neurotransmitters after we're done with them. See you then.